Hello students, how are you? I hope all of you are good and in good health. I am Ronia, your botany ma'am at MemoNeet and currently a final year medical student at Government Medical College, Mehboob. So as you all know, in this video, we are going to discuss about solutions of botany. But I hope before discussing solutions of botany, let me give you a tip. So whenever you are reading a question, read it carefully. Like let's suppose I am reading this question, I'm going to underline this. Read it carefully and do not forget this word, not. Okay, this not word is very important. It's extremely important and you should not forget this word. Okay, because if you, I see in exam is a pressure cooker situation. Exam is a pressure cooker situation, and in that situation your brain is under so much pressure, no, under so much pressure that it will take out things which are 100 times revised. Okay, it won't, if let's suppose you have seen one concept once in your life, that concept will not be remembered in exam, because under pressure cooker situations only the things which are revised nearly 100 times, those things only come up. Okay, so what happens under such pressure cooker situation? We tend to forget word not. You should not forget word not. Okay, so in a question, before answering, search for word not. If at all not is there, then read the question again. Okay, because not is a very dangerous word, very dangerous word. And this has happened with me so many times, and it still happens with me. So that's why I'm telling you, do not forget word not. Not is a dangerous word for an MCQ exam, I'm telling you. Very, very dangerous word it is. Okay. Yes. So now let us start. Question number 1. What does it say? Which of the following belongs to family Muscidae? Okay. Which of the following belongs to family Muscidae? The answer to this question is housefly, which is also known as Musca domestica. So the scientific name of the housefly is Musca domestica. It belongs to family Muscidae. Okay. Order Diptera. This is there in your NCERT under living world. Okay, you can go and take a reference. I have mentioned references in the answer document. It comes under class Insecta. Under phylum Arthropoda. See, little students, I want to tell you one thing, because when I'm explaining you, I am under a flow of teaching. Okay, so what happens? Sometimes I might make a little mistake in reading a question, or in forgetting the word, forgetting to see word not, as I told you. So never, do not hesitate and mention in comments. Okay, mention in comments. Okay, so I'll be there to clarify that. Yeah, so the answer to question 101 is housefly, that is Musca domestica. Which of the following belongs to family Muscidae? It's housefly, which is also known as Musca domestica. Family for your housefly is Muscidae, order is Diptera, class is Insecta, and phylum is Arthropoda. Okay, I hope it is clear to you. Okay, now moving on to question 102, naked seed plants are. See, they are asking that in these plants, seeds are present, but seed coat is absent. So these seeds, they become what? Naked, right? So what kind of, I would say, what kind of plants have seeds, but they do not have seed coat? Yes, can you remember? Do you remember? Tell me, do you guys remember? Yes, see, I'll tell you. Let us simplify this. So this says we know that two types of plants have seeds. That is gymnosperms and angiosperms. Both of these type of plants have seeds. In angiosperms, we know that seed has a seed coat, which is testa, tegmen, if you remember. Okay, but in gymnosperms, seeds do not have seed coat. Okay, that is why these seeds are naked. These seeds are naked. Now you will ask, why do seeds do not have seed coat? Because the seed forms from ovule, and if ovule is present inside ovary, then it will be enclosed. If ovule is not present inside ovary, then it won't be enclosed in a seed coat and these seeds will be naked. Understood? Now, because the ovules of gymnosperms are not enclosed by ovary, that is why these seeds are naked. Okay, this is a very characteristic feature of gymnosperms, that the seeds of gymnosperms are naked. So answer to this question is option number 4. Okay, that is D. Understood? Now let us see question 103. What does it say? In Pinus, if endosperm has 12 chromosomes, then how many chromosomes will be there in pollen grain? So you also know, I also know that endosperm, okay, first of all, you also know, I also know that Pinus is a gymnosperm. And in gymnosperms, endosperm is haploid. So now, if the pinus has endosperm and if it has 12 chromosomes, it means that the haploid number of chromosomes in pinus are 12. So now they are asking how many chromosomes will be there in pollen grain. Now, again, we all know that pollen grains are also haploid. So they will also have same number of chromosomes as that of pinus. Okay. So even pollen grain will have 12 chromosomes. So answer to this question will be, let us see, just a second. Yeah. So answer to this question will be option number 3, which says 12 chromosomes. Okay. I hope this was clear to you. Moving on to question 104, filamentous brown algae is. Filamentous brown algae means it's what does it mean? It means that this algae belongs to class Phaeophyceae and it should be filamentous. So what is an answer to this? Answer to this question is ectocarpus. Okay, so answer to this question is ectocarpus. Ectocarpus, just a second, belongs to class Phaeophyceae with Phaeophyceae means it's a brown algae. It's filamentous. It has pigments chlorophyll A, yes, C, yes, fucoxanthin. Okay, so answer to this question is option 3. Now, moving on. In cymose inflorescence, this is very easy question. In cymose inflorescence, peduncle has limited growth. The main axis terminates into a flower. The flowers are borne in acropetal order. Younger flowers are present towards apex. Now see, in cymose inflorescence is type of inflorescence in which the main axis of the plant body terminates into flower, and the flowers on the plant body are born in basipetal order. Okay. Basipetal order and not acropetal order. Basipetal order and not acropetal order. Okay. So it is saying flowers are born in acropetal order is wrong. Now, what do I mean when I say that in cymose inflorescence, flowers are born in basipetal order? What do I mean? I mean that the flowers which are older. See, let's suppose this is okay. So these are older flowers. Older flowers are present at apex, and young flowers are present at base. Okay. Do you understand? So basipetal order means that older flowers are present at apex and young flowers are present at base. And this kind of arrangement of flower is seen in cymose inflorescence. So what they're saying, young flowers are present towards apex. This is again wrong. This is again wrong. This seems correct. Yes, the main axis terminates into flower. This is correct. Peduncle has unlimited growth. Now I want to tell you one thing. If the main axis of the plant body is terminating into a flower, how will it have unlimited growth? Not possible, right? So even this is wrong. So answer to this question is option number B. Okay. I hope this was also clear. And if not, you know where to put your doubts in comments. So question 106, what does it say? When leaflets are joined together at a common point at the petiole, the leaf is, see, when leaflets are joined at a common point on a petiole, the leaf is known as, yes, palmate leaf. And are palmate leaf simple leaves? No, they are compound leaves. So palmately compound leaf. Okay. I hope this was clear to you. Now see, if this is a very simple question, direct bit from NCERT, this has to be clear for you. You cannot say that this was a very difficult question. I would rate it 1 by 10 on the level of difficulty scale. So this is an extremely easy question. Okay. Now, before moving further, I want to clarify a few things. See, peduncle, we were reading in before question, right? Peduncle is axis or, okay, stalk of flower. Okay. So peduncle is stalk of flower. Okay. And petiole is stalk of leaf. Okay, this is very basic thing which I think you should know. If you don't know, it will be a very big blunder for you, all of you guys. So that is why I thought, let me clarify. Okay, so this was your question 106. Moving on to question 107. Let us see what does it say. It says ring arrangement of vascular bundle is a characteristic feature of. Now, whether dicot or monocot, we do not have to think about that. First, we should know ring arrangement of vascular bundles is seen in roots or in stems. Yes, it is a very obvious thing that ring arrangement of vascular bundles is not seen in roots. So root वाला ऑप्शन तो निकाल देना चाहिए. ठीक है, अब हमारे पास दो है, monocot stem, dicot stem. So now you have to use your memory and remember, In which type of stem ring arrangement of vascular bundle is seen? Yes, it is seen in dicot stem. Okay. And as you all know that in monocot stem, the vascular bundles are scattered. Okay. Something like in ground tissue of the stem. So these are the vascular. Okay. Now worst drawing I'll do. See, these are the scattered vascular bundles. Okay. Yes, so now we will move on to question number 108. What does it say? A dorsiventral leaf is identified by presence of, see, dorsiventral leaf is a dicot leaf, and in a dicot leaf, there are difference of stomata present on the upper and lower surface. Okay, and the upper surface of the leaf of a dicot leaf has less stomata, and the lower surface of dicot leaf has more stomata, and the upper surface is known as a daxial surface. Lower surface is known as a baxial surface. Okay, this you have to remember. Upper surface is known as a daxial surface, and lower surface is known as a baxial surface. Okay, and where are more stomata present? On the lower surface, so as to prevent transpiration, because during daytime when lot of sunlight is there, all the sunlight will fall on upper surface of the leaf. So if upper surface of the leaf is having more stomata, all the stomata will open and there will be more transpiration, more loss of water. Okay, it is not beneficial to plant. That is why more stomata are present on lower or a baxial surface. Okay. So the answer to this question is option C, which says a baxial epidermis bears more stomata than a daxial epidermis. A dorsiventral leaf is identified by presence of stomata on both the epidermis. See, even the monocot leaf, dorsiventral means dicot leaf. Even the monocot leaf, isobilateral leaf, has stomata on both the epidermis. So you can't differentiate a dorsiventral leaf and a isobilateral leaf just on the basis of presence of stomata. Okay, now a daxial epidermis is having more stomata. It is saying that the upper surface of the leaf is having more stomata. Again, this is wrong, because if upper surface is having more stomata, more transpiration and more water loss will occur. Okay, and what is fourth option saying? It is saying no stomata in epidermis. This is a big blunder. Leaf has stomata in epidermis. Okay, so again, the option 3 is the correct answer to question 108. Question 109. Which of the following organelles constitute an endomembrane system? The organelles which constitute an endomembrane system are lysosome, vacuole, Golgi, ER, and all of these organs are bound by single membrane. So, let us see whether this question is having not or not. Which of the following organelles constitute an endomembrane system? A only, A, 9. So D is the correct answer. Okay. Question 110. What does question 110 say? Let us see. Saying the following features belong to which type of fungi? Yes, very interesting. Let us read. The following feature belong to which type of fungi? Mycelium is septate and branched. Okay, so we have mycelium, which is branched. Mycelium, which is branched. And septate. Okay. Some members are saprophytes, or okay, so we have parasites also in this fungi. And this is Deuteromycetes. And they reproduce only by conidia. Okay, so they reproduce only by conidia. So asexual method. Of reproduction is only asexual spores, which is conidia. So now, what do you think these fungi can be? Okay, so these are the statements directly taken from NCRT. If you see the deuteromycetes paragraph in your NCRT, you will see that mycelium is mentioned to be septate and branched. The members are mentioned to be saprophytes or parasites, or they help in decomposing litter or mineral cycling. Oh, I'm sorry, just a second. Or mineral cycling. An example for these fungi are trichoderma, coletotrichum. And what are these all characteristics of it? They are characteristics of deuteromycetes. Okay. Now, one thing I want to tell you all guys, trichoderma is a very important pathogen which causes multiple plant diseases. Okay. And this is an important question from your chapter of microbes and human welfare in class 12. Okay, so moving to question 111. Inulin is a polymer of, yes, fructose. See, this is the simplest question, and I'm very happy to see this question also because, come on, this is the easiest question. Okay, inulin is a polymer of fructose. And I want to tell you an extra edge thing here. Inulin, when you'll come to MBBS, you'll read that inulin is used to calculate glomerular filtration rate. Glomerular filtration, if you have ever read your excretory products chapter in zoology, there you will read that there is something called glomerular filtration rate, GFR. Okay, so inulin is used to calculate GFR. Okay, why? Because inulin is not metabolized in body. Okay, so how you are taking inulin, that way only it has come out of the body. There is no metabolism or no change occurring to this polymer inside the body. Okay, that is why this is used in calculation of GFR. Okay, so starch forms helical dash structure that can hold molecules and gives blue color. So how is starch forming? Starch forms helical dash structure that hold molecules and give blue color. Okay, what molecules will starch hold? It will hold iodine molecules. And what structure does it form? It forms secondary structure. Okay, so starch, wait, I'll tell you. Holds iodine molecules in its structure, helical structure, and this gives blue color. Okay, I hope this was clear to you. So secondary structure of starch as a helical, here iodine is trapped and it gets blue color. So when you add starch solution to iodine, iodine will get trapped in these helices here and it will give blue color to the solution. Okay. So secondary structure of starch gives blue color. Cellulose cannot give. Okay. Why? Because cellulose does not have helical structure. Okay. I hope this was clear to you. So answer to question 112 is option number 2. Yes, so we will now see question 113. Let us see what is it saying. It is saying which of the following is chief pigment associated in photosynthesis. Now, this is an extremely simple question. The chief pigment associated in photosynthesis is chlorophyll A. Okay, majority of the light required for light reaction to synthesize ATP, and NADPH is captured by chlorophyll A. Okay, majority of light required to synthesize ATP and NADPH during light reaction is captured by chlorophyll A. That is why chlorophyll A is the chief pigment during photosynthesis. Okay, I hope this is clear to you. So answer to question 113 is option 1. Okay, it's option 1. Moving on to question 114. Which of the following is formed in C3 plants during photosynthesis? In C3 plants, during photosynthesis, 3-phospho. Okay, let us see. In C3 plants, there are 3 things, 3 steps which occur during photosynthesis. These or during Kelvin cycle. 3 steps that occur during Kelvin cycle. The first step is carboxylation, and during this step, carbon dioxide combines with RuBP, ribulose 1,5 bisphosphate to form 3-PGA. Okay, what is 3-PGA? 3-phosphoglyceric acid, otherwise known as 3-PGA. Now I want to clear one thing here, that see, phosphoglycerate and phosphoglyceric acid is the same thing. Okay, so like ric acid, glyceric acid or glycerate, you can use anything. Okay, for example, I'll give you alpha ketoglutarate or alpha ketoglutaric acid. So this you will notice during biochemistry in your first year MBBS after you get your MBBS seat. So you'll notice these things. So remember, I'm telling you now only. Rate is ic acid. Rate, suffix rate can be replaced by suffix ric acid. Okay, important. Out of your little extra edge. But remember, okay, so this is the first step of Kelvin cycle. What is the second step of Kelvin cycle? Reduction. Okay, second step of Kelvin cycle is reduction, and the third step of second step of Kelvin cycle is reduction. During this, ATP and NADPH synthesized during light reaction is utilized. Okay, and third step is regeneration. During this, the ATP which is utilized is resynthesized. Okay, I hope this is clear to you. Kelvin cycle is also known as what? Dark reaction. Okay, and these occur in stroma of what? Chloroplasts. I hope this is clear. So phosphoglyceric acid is the correct answer to question 114. Okay. Now moving on to meiosis has evolutionary significance because it results in 4 daughters. No. Genetically similar daughters? No. If at all meiosis was ever resulting in genetically similar daughters, it would never have had evolutionary significance only. So what is it? I'll tell you. See, during meiosis, okay, there's something called crossing over. So let us suppose this is mother chromosome, this is father chromosome. Okay, so wait, I'll tell you. See, this is mother chromosome. This is father chromosome. After crossing over, what happens? This happens. A segment of DNA is exchanged between mother chromosome and father chromosome. See, so this is after crossing over, and this exchanged DNA now this after exchange of DNA between the mother chromosome and the father chromosome, there is the new chromosomes that are formed are unique and give unique features to the organism that will be synthesized from this chromosomes. Okay, or that will receive these chromosomes. Okay, so that is why due to crossing over, there is exchange of genetic material between mother and father chromosome. This helps in synthesizing new unique chromosomes and which are transferred to the new generation. That is why the new generation is also unique, not the exact carbon copy of parents. Okay, now see, meiosis is seen in human beings also. Okay, like it is seen in human beings also. Now if you remember the human reproduction chapter of your class 12, we know that spermatogonia undergo meiosis to form sperm, and uh, oogonia undergoes meiosis to form ovum. So because of the meiosis, we are not carbon copy of our parents. If at all meiosis did not happen, we would have been carbon copies of our parents. Okay, and this process of this total process of crossing over and synthesis of new unique chromosomes and generation of new unique organism is known as recombinations. Okay, recombination. And this is the unique feature of meiosis, which is helped in evolution. Okay, now how it has helped in evolution? See, now our parents have some faults. Okay, have some faults. If at all we would have been carbon copies of our parents, we would also inherit those faults, right? Those faults. But because we are not carbon copies of our parents, we have not inherited those faults. Okay, are you getting me? I am saying that there are certain problems. No, I would say there are certain characteristics in our, okay, let me put it in a very nice English. I'll tell you, there are certain characteristics in our parents which are not favorable to their survival in earth. Okay, to their survival in normal. There are certain characteristics in our parents which are not favorable to their survival. Okay, now because we are not carbon copies of our parents, we do not have those characters. Okay, are you getting me? If we would have been carbon copies of our parents, we would also have those characters which are not favorable for survival. Are you getting me? So we have evolved. Now, why we have evolved? Because of meiosis only. If meiosis would have not happened in our parents, we would also have those characters, right? So because of meiosis, we have evolved from our parents, and we have our own set of unique characters. And what happens during meiosis? Recombinations happen. Okay, so meiosis has evolutionary significance because it results in recombinations. Okay, I mean, I really want to explain you guys more in depth because that's what I love doing. But I don't think the time will ever permit us to just discuss what is meiosis. Okay, so now coming to this, Blackman's law of limiting factor is applied to. Yes. Now, if you can remember this, then you are a great person because this is the only named law which is present in your total uh, question paper. In this question paper, this is the only named law you will find. Okay, so this is found in respiration. This is found in respiration. That is what I am saying. Let me see in comments how many of you are telling I am wrong. Okay. Yes. So the fourth option is photosynthesis. Yes. Blackman's law of limiting factor is found in photosynthesis. Related to photosynthesis. Now, let me tell you what does it say. It says that, I'll tell you what does it say in the end, but now let us just listen to me very, very carefully. I want your attention on me. Okay, now see, we know that photosynthesis requires, we know that photosynthesis requires ATP, NADPH, carbon dioxide, sunlight, water. For it requires all these things to occur. Okay, photosynthesis requires all these things. Means these all are the factors which affect the rate of photosynthesis. Isn't it? Matlab, what do I want to say is all of these are the factors which will affect whether the photosynthesis is occurring fastly or slowly. If at all one of them is even decreasing or increasing, it will affect the rate at which photosynthesis is occurring. Isn't it? It will affect the speed of the photosynthesis. Instead of speed, I am using the word rate here, isn't it? So all of these are the factors which affect the rate of photosynthesis. Okay, now listen to me very carefully. Among all these factors, the factors which is present in very limited amount, the factor which is present in very limited amount will be the most important factor, and that factor will primarily decide the rate at which photosynthesis is occurring. Are you getting me? So this total thing, whatever just now I have told you, is told by Blackman. Even I told it, but nobody told it Ronia's law. Okay, funny. But yeah, so Blackman told this thing. Okay, what did he say? Let me tell you. He told that according to Blackman's law, in a biochemical process. See, photosynthesis is a biochemical process because it's a chemical process occurring in a biological being, in a living being. Okay, that's why it's a biochemical process. So Blackman told that, and I state, in a biochemical process, if multiple factors are affecting its rate, then the factor which is present in the most limited amount will be deciding its rate primarily. Okay, will be deciding its rate primarily or will be affecting the rate directly. Understood? So this is what Blackman said, and I am repeating sadly. Okay, because it would have been good if it was said Ronia's law, but nobody's saying it. So sad. I hope there will be one day. Where the umeed kayam hai. So moving on to, so answer to this question is option number 4. Okay, moving on to question 117. What is it saying? It is saying given below are two statements. One is labeled as assertion and the other is labeled as reason. Okay, and choose the correct answers after reading the assertion and the reason. Okay, so let us see. The assertion says that proton gradient is important because breakdown of this gradient leads to release of energy. Proton gradient is important because breakdown of this gradient leads to release of energy. Okay, this is correct. How? I'll tell you. ATPase has a channel that does not allow diffusion of proton back across the membrane. Okay, see. I'm sorry. So see, this basically is stating chemiosmotic hypothesis. Okay, so according to chemiosmotic hypothesis, let us suppose this is your thylakoid, one thylakoid. Okay, in chloroplast we have thylakoids, right? Coin-like structures. So let us suppose this is one thylakoid. Okay, I have extreme level of bad drawing. And that is the reason I do not like anatomy, which is your subject in first year of MBBS. I hate that subject. But that does not mean you also have to hate it. You can love it. Okay. Will I be ever able to draw a thylakoid? Let us see. Yes, I will be successfully. I'm able to draw it. So see, Never mind. See, this is a thylakoid, and these are the thylakoid membranes. Okay, this is inner membrane, this is outer membrane. Okay, and in the membrane we have this B6F cytochrome. Okay, here you have PS1, photosystem 1. Here you have PS2, police station name, jokes name, photosystem, PS2. Okay, and here you have your ATPase enzyme. Okay, so during cyclic and non-cyclic photophosphorylation that occurs between photosystem 1 and photosystem 2, what happens? There is accumulation of H plus ions inside the lumen of the thylakoid. Okay, so this due to this, what happens? There is high concentration of H plus ions inside the lumen of the thylakoid. Okay, this high concent, because there is high concentration of H plus ions inside the lumen of the thylakoid, these H plus ions, they move down the gradient, that is, they move from their high concentration to their low concentration, which is there here outside in the stroma. Okay, through this ATPase. Okay, and this movement generates energy. This movements generate energy, which energy is used to convert ADP, adenosine diphosphate, to ATP, adenosine triphosphate. Okay, are you getting me? So this is what occurs. This is what is told by this is what is chemiosmotic hypothesis in short. Okay, so now what it is saying, proton gradient is important because breakdown of this gradient leads to release of energy. Yes. See, what is gradients? The gradient is a term used for difference in concentration of a substance across a membrane. Okay, so now these are the membranes. Okay, see, what I am saying is, now these are the thylakoid membranes, and there is difference in concentration of H plus ions between the lumen of the thylakoid and the stroma of the chloroplast. Are you getting me? And this difference is known as proton gradient, and it is important for generation of energy, because if this gradient is not present, the H plus will not move out. Okay, understood? Are you getting me? So this statement is correct because it is saying that proton gradient is important to allow. Proton gradient is important because breakdown of this gradient leads to generation of energy. When does this gradient break down? When the protons are moving from the lumen to outside, this gradient breaks down. Okay, so assertion is correct. Reason says ATPase has a channel that does not allow diffusion of proton back across the membrane. This is wrong. Okay, the protons, they can diffuse across the ATPase back into the lumen. Okay, understood? So the answer to this question is assertion is correct, but reason is no, incorrect. So answer is second option. Okay. What does 118 say? Let us see. Match list 1 with list 2. Complex 1, cytochrome oxidase. So you have to match different complexes. It is very easy question. Okay. Let us see. First, I'll put this because again, it is important to, put this also. I don't know how to, just a second. So answer to question 117 was, answer to question 117 was option number 2. Okay. Yes. So these are the complexes which are involved in electron transport chain, which occurs after Krebs cycle in during respiration. Okay, so our complex 1 is NADH dehydrogenase. Okay, so complex 1 is known as NADH dehydrogenase. Complex 2 is known as succinate dehydrogenase or FADH dehydrogenase. Succinate dehydrogenase or FADH dehydrogenase. Okay, that's another name. Complex 3 cytochrome BC1. Complex 3 is cytochrome BC1. So this is C. Okay, and complex 4 is cytochrome C oxidase. So we have A3B4 C2. A3B4 C2 D1. A3B4 C2 D1. Option 2. Okay, moving on to, so let us add explanation here. See, complex 1. Okay, I'll add later on. Okay, it will be a very late thing if I add now only. So yes, moving on to question 119. What is redifferentiation? Yes. So there are three terms in plant growth: differentiation, dedifferentiation, and redifferentiation. Okay, differentiation is a process during which plants lose their ability to divide and they gain a specialized function. Okay, so differentiation is loss of ability of a tissue to divide after gaining a specialized function. Okay, dedifferentiation is regaining the capacity of division by the tissue which has already undergone differentiation. Okay, what is redifferentiation? Redifferentiation is losing the capacity to divide again after regaining the capacity to divide, that or losing the capacity to divide after dedifferentiation. So what is that? That is redifferentiation. Okay, so losing the capacity to divide after regaining it through dedifferentiation. Okay, that is redifferentiation. Very simple it is. I mean, it looks complex. So let us move on to 120 question. What does it say? It says that which of the following hormones plays an important role in seed development, maturation, and dormancy? So it is asking that which of the following hormones, that is, which plant hormones that we know, that is auxins, cytokinins, ethylene, abscisic acid, which among these is playing a role in development, seed development and seed dormancy? Yes, so seed, as you also know, I also know that seed development, seed maturation, and seed dormancy, all of these are special functions of abscisic acid. Okay, all of these are special functions of abscisic acid. Abscisic acid also helps during stressful conditions. Therefore, it is known as stress hormone. Okay. So, answer to question 120 is option number 1. I hope it is clear. Yes. So, moving on to question 121. Ovule is a small structure attached to placenta by means of a stalk. So, see, ovule is present inside ovary, and in ovary it is attached to placenta with the help of a stalk which is known as funicle. Okay, so this is the ovary. Inside it, here ovule is attached with the help of a stalk, and this is known as funicle. Okay, so answer to question 121 is option number 3. Moving on to question 122. What does it say? The coconut water is 3 nuclear endosperm. Okay, now see, There are certain cells in which after nuclear division, that is when the nuclei have divided, after nuclear division, there is no cytoplasmic division. Okay, so if in a cell, nuclei has divided into 2 or 3, okay, but the cytoplasm is not dividing, it remains as it is, then what happens, that single cytoplasm will have multiple nuclei, right? It will have multiple nuclei. And this condition, my friends, with one cytoplasm having multiple nuclei is known as syncytium. Syncytium or multinucleate condition. Multinucleate condition. And why does this happen? It occurs when nuclear division, that is karyokinesis, is not followed by cytokinesis. Okay, so that is why the endosperm, which is formed by this process, is free nuclear, because in endosperm, that is in cytoplasm, the nuclei, multiple nuclei are lying freely. Okay, free nuclear endosperm. Okay, in this, what happens is, during the fusion of central cell with male gamete, central cell with male gamete, the central cell has one cytoplasm and 2 nuclei. So during the fusion of central cell with one gamete, there is karyokinesis occurring, but there is no cytokinesis. Okay, so this generates a multinucleate condition, giving rise to free nuclear sperm, for which is coconut water. Okay, so this was question 122. Moving on to question 123. What does it say? Which enzyme produced by microbes is an important ingredient in detergent formulations? So we use detergents for washing clothes. Okay, for washing clothes. So now, mainly clothes have stains of fats, like oils, ghee, sabzi also has oil. So again, in our clothes, there will be stains of oils, and we want to remove that stains mainly. So that is the primary purpose of washing clothes. So now we all know that oils are fats, and fats are broken down by enzymes called lipases. They break down fats into fatty acids and glycerol. Okay. So lipases are the enzyme which break down fats into fatty acids and glycerol. And when the fats are broken down into fatty acid and glycerol, they are removed. The stain gets removed, because stain is due to oil, the fat, the whole molecule of fat. When we are breaking it, the stain also gets removed from the cloth, and this helps in cleaning the clothes. Okay, and this is done by enzyme lipase, and this is present in detergent formulations. Okay, and this question is from your chapter Microbes in Human Welfare. The part of prime, so this was, so answer to question 123 is option number 2. Moving on to question 124, it is saying the part of primary treatment of sewage that is taken for secondary treatment is, yes. So see, during primary treatment, the sewage is put into a primary treatment tank. Okay, primary treatment plant. And during that, there is settlement of heavy particles down. Okay, settlements of heavy particle down. And the light particles, they come out and they come up and float. And this is known as supernatant, which is also known as effluent, and it is put in Secondary, it is put in secondary treatment plant. The effluent which is floating on the upper surface of the primary treatment tank, which is also called supernatant, is taken to secondary treatment tank. Okay, understood. So secondary sludge is not the answer, activated sludge is not the answer. Yes, the answer to this question is effluent. That is option number 3. Okay, I hope it was clear to you. So answer to question 124 is option number 3. Okay, so this is what happens. So now the effluent is put into the secondary treatment plant. So moving on to question 125, what does it say? Let us see. It says sickle cell anemia is caused by. So yes, sickle cell anemia is caused by replacement of glutamic acid with valine in globulin. Yes, that is a correct statement. It is a correct statement because in sickle cell anemia, so hemoglobin has 2 alpha chains and 2 beta chains. So in sickle cell anemia, in beta chain at 6th position, normally there is glutamic acid. Okay, but in sickle cell anemia, the glutamic acid is replaced by valine. Okay, and why does this happen? Because of change in one single base pair. Okay, that is GAG is changed to GUG, and this change causes replacement of glutamic acid with valine. So when glutamic acid is replaced with valine, what happens is the RBCs, which are normally biconvex, they become sickle shaped. So the RBCs, which are normally biconvex, they become sickle shaped in sickle cell anemia. Now, because of this sickle shaped RBCs, the ability of the red blood cell to carry oxygen to different tissues decreases, and this causes hypoxia. That is a state, hypoxia is a state of decreased oxygen in the body, and this causes all the symptoms of anemia like breathlessness, weakness, dizziness, headache, all of it. Okay. So this statement is correct that sickle cell anemia is caused by replacement of glutamic acid by valine in beta globin chain of hemoglobin. It's a correct statement. Let us read next statement. What does it say? It says caused by change in multiple base pairs. No, I just told you that it is caused by change in one base pair only. That is GAG is changed to GUG. So A changed to U. Characterized by biconvex RBCs, yeah, that is incorrect. Sickle cell anemia is characterized by sickle-shaped RBCs. Okay, I hope it is clear. And the fourth option says that it's an autosomal dominant trait. Now, this is to definitely a very wrong option because sickle cell anemia is an autosomal recessive trait. Okay, so answer to question 125 is option number 1. I hope that is clear to you. Moving on to question 126. This is a very easy question, direct line from NCERT. What it is saying, the frequency of recombination between gene present on the same chromosome, okay, as measured as a measure of distance between gene was explained by Alfred Stewart. Okay, so this is a direct statement from NCERT. Your NCERT says Alfred Stewart explained the recombination frequency between genes present on the same chromosome. Okay, based on the distance present in genes. So what it is saying that there is a chromosome. In this chromosome, there are 2 genes present. Okay, and in these chromosomes, there are 2 genes present. Now, Now, what is the frequency with which there will be recombination in these two genes will depend upon the distance present between these two genes. And this was explained by Alfred Stewart. Okay, so answer to question 126 is option number 4. Moving on to question 127, what does it say? Match the following with respect to nucleic acid. So 5' to 3' bases in RNA, 5' to 3' bases in RNA. Okay. Sorry, 5' to 3' different bases in RNA. 3.4 nanometer polarity of DNA or RNA. 3.4 nanometer of DNA, pitch of S. So let us see. Yes. So here, can you see? See, 5' to 3' is polarity of DNA or RNA. Polarity of DNA or RNA. That means that when you see a DNA or RNA strand, the one end of a DNA strand will be 5' and the other end of the DNA strand will be 3' and it is the same in RNA also. So option B is 1. Okay. Now, 3.4 nanometer is pitch of DNA. Okay, 3.4 nanometer is pitch of DNA. That is, okay, so that has to be the option number 5. Here you can see. If you see, option number 5 is polynucleotide chain in DNA. Okay. Yes. So 3.4 nanometer is pitch of helix. That is second option. So what is pitch of helix? When you see your DNA, there is one coil, right? Like this. This distance of total one coil of DNA is pitch of DNA helix. Okay, is pitch of DNA helix and it is 3.4 nanometer. Okay, I hope that is clear to you. Moving on to 0.3 nanometer distance between base pairs. Distance between base pairs. So 3.4 nanometer is, sorry, 0.34. 0.34 nanometer is distance between base pairs. So this is option 3. So what does it mean? It means that when you see in a DNA, see, in a DNA, when you see, this will be adenine, thymine, guanine, cytosine. Okay, distance between these two pairs is known as, these two base pair is 0.34 nanometer or in other terms 3.4 angstrom. Okay, 1 angstrom is 10 to power minus 10. Understood? So, distance between base pairs is 0.34 nanometer. Okay, now there are two polynucleotide chains in DNA. So, there are how many polynucleotide chains in DNA? See, this is one chain, this is another chain. So, two polynucleotide chains in DNA. Okay. So, answer, so two polynucleotide chains in DNA is E is 4. Okay. Understood? E is 4. And yes, there are 4 different bases in RNA. 4 different bases is RNA. So A is 5. 4 different. What are those? Adenine. Yes. Uracil. Yes. Guanine, cytosine. These are the 4 different bases present in RNA. So answer would be yes. 1B, 2C, 3D, 1B, 2C, 3D, 4E, 5A, 1B. Let us see. 1B, polarity of DNA and RNA. 1B, 2C, 3D, 4E, 5A. So answer is option number 4. So answer to question 128 is option 4. This was, for me, I think this is a very easy question, but it can be a difficult question for all of you guys. So Don't worry, just read NCERT carefully. This is, I guess, a very easy question. Moving to 128, question number. What does it say? It says chromosomal composition of Klinefelter syndrome is, yes. So Klinefelter syndrome may we have something like, yes, 40. We have two X chromosomes. Okay. And we have 47 plus XXY. What do we have in Klinefelter syndrome? 47 plus XSY, because normally we have 46 chromosomes, but in this there is one extra X chromosome, so total number of chromosomes become 47, and we indicate that it has two X chromosomes, so we write XXY. Okay, I hope it is clear to you. This was very easy question. I would rate it as 2 by 10. Okay, so answer to question 128 is option 4. Moving on to 129, during transcription, the site at which RNA polymerase bind at DNA is called, yes, promoter. So what it is saying, during transcription, the site at which RNA polymerase binds to DNA, what it is called? It is called as promoter. So site at which RNA polymerase binds to DNA is nothing but promoter. Okay. I hope it's clear to all of you guys. Yeah. Now moving on to, so answer to question 129 is 1. Moving on to question 130, what does it say? Altitude sickness includes heart palpitation, fatigue, nausea, all of this. See, altitude sickness is nothing but when we go to high mountains, what happens is there is increase in heart rate, which is felt by us. So that is known as heart palpitations. Okay, there is nausea because as we go up, as we go higher in the mountains, there is decreased oxygen supply to tissues. Okay, this decreased oxygen supply is to tissues is known as hypoxia, and this stimulates a center called chemoreceptor trigger zone, chemoreceptor trigger zone, CTZ, in your brain. Okay, and triggering of chemoreceptor triggers zone causes nausea. Okay, understood. And there is fatigue. Why is there fatigue? Because for our body to work, it requires oxygen. But as we move up mountains, there is decreased oxygen supply to different tissues present in the body. So because of decreased oxygen supply, there is increased in tiredness, that is fatigue. So all these three things are experienced by a person who is climbing up a mountain suddenly. Okay. All these things are experienced by a person who is climbing up a mountain suddenly, when he is not giving his body time to accommodate to the change in conditions. Okay. Then all these things occur, and these things are known as altitude sickness. Okay. I hope it's clear to you. So answer to question 130 is option number 4. Moving on to question 131. Find the correct statements. Okay, so we have statements, it seems, and we have to find correct one among those. So we have 4 statements, A, B, C, it seems. So there is a mistake here. Never mind. The statements are A, B, C, but answer is given in the form of 1, 2. So it's a printing mistake. Ignore it. The diversity at the species level, for example, the Eastern Ghats have greater amphibian species diversity than the Western Ghats. Okay. So let us read. In every video I give you, I told you to read not properly. Now, in this video, I am telling you read correct and incorrect properly. So we have to read whether the question is asking correct or incorrect. So this question is asking correct statement and not incorrect statement. Okay. So what is it saying? This is saying the diversity at the species level. For example, the Eastern Ghats have greater amphibian species diversity than the Western Ghats. See, no, Western Ghats are hotspots. Okay. So they have greater diversity than Eastern Ghats. So this statement is wrong. Okay, I hope that is clear to you. According to International Union for Conservation of Nature and Natural Resources, IUCN 2004, the total number of plant and animal species described so far is slightly more than 1.5 million. Yes, this is correct. This is a direct NCERT statement, exact carbon copy. If you open your NCERT, you will see the statement exactly. Okay, even is not changed here. Okay, understood. Now moving on to statement 3, what does it say? It says biomass of species is expressed in terms of fresh weight only. Biomass of species is expressed in terms of fresh weight only. It's incorrect. The statement given here is incorrect. Okay, biomass of species is not expressed only in terms of fresh weight. So for measuring biomass, we also include dry weight and not only fresh weight. So even dry weight of species can be used to measure biomass. Okay, I hope that is clear to you. So the statement is wrong because only is written. Now moving to statement number 4, what does it say? For calculation of energy contained at a trophic level, all organisms belong to that trophic level are not included. No, this is an incorrect statement because for calculation of energy at a trophic level, trophic level means a level in a food chain. So for calculation of energy at a trophic level, all organisms at that level are included. Okay. So this is also an incorrect statement. So answer to this question is option number 2. Okay, I hope it's clear to you. If you have any doubts, you know where to put them. Now, moving to question 132. What does it say? What is the Evil quartet? Yes, what is the Evil quartet? So Evil quartet refers to 4 things which cause biodiversity loss. Do they cause mass extinction area? No. Do they cause pollution? No. Do they cause most polluted place based on Earth? It's actually place. It's place, spelling mistake. So this is also wrong. What is Evil quartet? Evil quartet refers to 4 things which cause biodiversity loss. Okay, they cause accelerated loss of biodiversity. Okay. Yes. So moving on to what is Evil quartet? The Evil quartet is nothing but your loss of habitat and fragmentation, overexploitation of species, alien species invasion, and last but not the least, extinction of species. Okay, so what does co-extinction of species mean? It means that if one species is getting extinct, the other species will also get extinct. Okay, I hope that is also clear to you. So there are 4 things which occur in Evil quartet. The first one is loss of habitat. The second one is overexploitation. The first one is loss of habitat and fragmentation. The second one is overexploitation of species. The third one is alien species invasion. And the fourth one is co-extinction of species. Okay, so these are the 4 Evil quartet. Moving on to question 133. What does it say? Which of the following is not salient feature of genetic code? So which of the following is not a salient feature of genetic code? The code is triplet, it's correct. Genetic codes are non-ambiguous. They are not ambiguous. They are not ambiguous. This statement is incorrect. It is exactly it is universal. Yes, the genetic codes are universal. Okay, that is also correct. So both that is also correct. So but here what it is written, this is written exactly universal. Exactly. Okay, so they are not exactly universal, but they are universal. Okay, they are universal. They are not exactly universal, but they are universal. So we have to answer which of the following is not salient feature. Not. Okay, not. So this is also genetic codes are non-ambiguous. So this is not a salient feature and they are universal, but not exactly universal. Okay, so this is also incorrect. So answer to this is option number 4. Now, why is genetic code not exactly universal? Because not exactly universal because They are not same everywhere in the world. There are some changes which are encountered, but almost they are similar. Okay, means if AUG is coding for methionine, it will code for methionine in almost all organisms, but there might be some changes. Okay, so they are almost universal, but not exactly universal. Moving on to question 134, what does it say? It says that in gymnosperms, megaspore forms. Okay, so moving on to question 134, what does it say in gymnosperms? Megaspore forms haploid endosperm. Yes, it forms haploid endosperm. Megaspore endosperm in gymnosperms is formed before fertilization. So in gymnosperms, megaspore forms haploid endosperm. Female gametophyte, yes, it forms female gametophyte and female sex organs. So it forms all of these. That is why all these options are incorrect. All these options are not the correct answers because all of these are true. So single one cannot be the correct answer. So answer to this question is option number 4. That is fourth option. Question 135. What does it say? Important characters of tapetum are. Important characters of tapetum are. Nourishes the egg. Yes, it nourishes the egg. Middle layer of, is tapetum middle layer of megasporangium? No, it is not middle layer of megasporangium. Okay, is it multinucleate? Yes, it is multinucleate. So answer to question 135 is option 4, that which is saying 1 and 3 are correct answers. Okay. Moving on to question 136, what does it say? Identify incorrect statements regarding bacteria. So we have to identify incorrect statement regarding bacteria. What does it say? It says bacteria are most abundant microorganisms. Bacteria can live in extreme habitats where very few life forms can survive. Yes, archaebacteria can live in extreme habitats where very few life forms can survive. Okay. Very few life forms can survive, and they can be halophiles which live in salty areas. Okay. They can be thermoacidophiles which live in high temperature and acidic areas. Okay. I hope that's clear to you. Bacteria as a group show most extensive metabolic diversity. Bacteria as a group show most extensive metabolic diversity. Okay, this is also a correct statement. Okay, but there are multiple types of bacteria you can encounter. So this is also correct. Bacteria are most abundant, 100% they are most abundant, and they are obligate intracellular parasites. They are not all. There are bacteria who are obligate intracellular parasites, but not all bacteria are obligate intracellular parasites. Okay. So this option is incorrect. So answer to question 136 is option number 4. Now moving on to pick the odd one out. Okay, so we have to pick which is not correct. Okay, so Cycas is dioecious. Yes, because male and female gametophytes are present on different plants. So this is correct. Salvinia is heterosporous. Yes, it is correct because it produces megaspores and megaspores and microspores. Equisetum is homosporous because it produces only one type of spores. It is correct. Okay. Pinus is dioecious. No, Pinus is monoecious. Pinus is monoecious. In Pinus, the male and female gametophyte are present on the same body. So Pinus is not dioecious, it is monoecious. So answer to question 137 is option number 4. I hope that is clear to you. Moving on to question 138. What does it say? It says answer to question 137 was option number 4. Moving on to question 138, what does it say? It says which of the following are likely to be present in deep sea water? Okay, so which of the following are likely to be present in deep sea water? The answer is archaebacteria. Okay, in deep sea water, there are extreme conditions, extreme environments, extremes of conditions like acid, like highly acidic conditions, faulty areas. So in those extreme conditions, archaebacteria are known to survive. Okay. I hope that is clear to you. And why do archaebacteria can survive such extreme and harsh conditions? Because they have branched chain of lipids in their cell membrane. Because they have branched chain of lipids in their cell membrane. Okay, so I hope this question is clear to you. So answer to 138 is option number 4. Moving on to 139, what does it say? Tricarpillary. It says tricarpillary syncarpous condition is found in which family? So yes, tricarpillary means there are 3 carpels, and syncarpous means the carpels are fused. So this kind of condition is found in Liliaceae family. So answer to question 139 is option 1. This question, see, Liliaceae family has been removed from latest syllabus of NEET 2024, but this question is there for your practice. Okay. So I wanted to inform you that point. Now moving on to question 140, what does it say? The perianth is found in which flower? The perianth is found in a flower in which calyx and corolla are not distinguishable. Stamens are leaf-like, corolla leaf-like. See, perianth of a flower is a state, or sorry, is a condition where calyx and corolla are not distinguishable. Okay. So answer to question 140 is option 1. Bulliform cells are found in leaves of, bulliform cells are found in leaves of monocot plants. So we just have to know that which among the following are monocot plants. Okay. I hope that is clear to you. We just have to know that which of the following. So sunflower is a dicot. Okay. Sunflower is a dicot. Nerium is a dicot. Grasses are monocots. Lotus is dicot. Okay. So this question is very easy because even if you don't know that sunflower, nerium, and lotus are dicots, you will know that grass is a monocot. That's, if you don't know that also, I suggest you read NCERT once again, because grass is a monocot is a big example. Okay. So even if you don't know that sunflower, nerium, and lotus are dicots, you know that grass is a monocot. Okay. So in grasses, bulliform cells are found on adaxial surface. Okay, adaxial surface. And what are bulliform cells? Bulliform cells are nothing but large cells. Okay, so bulliform cells, see, there are some epidermal cells which get modified to form large colorless cells which are empty, and these cells are called bulliform cells. Okay, what they are called? Bulliform cells. Okay, so I hope that is clear to you. So answer to question 141 is option number 3. Okay. So what do these bulliform cells do? So what happens is these bulliform cells are empty. Okay, so that means they can absorb water. So when these bulliform cells absorb water, they become turgid, and leaf surface become, when they absorb water, they become turgid. That is, they become, turgid means hard, and it exposes the surface of the leaf. When the water is not present inside the bulliform cells, they are empty, they are flaccid, they become flaccid, and the leaf curls. Okay, so grasses open when there is water in bulliform cells, and they curl when there is no water in bulliform cells. Okay, so answer to question 141 is third option. Moving on to question 142, what does it say? It says match the following, and we have 2 columns. Let us see what are those. We have 2 columns. So we have names of scientists and what did they do. Okay, so we have names of scientists and what did they do. So let us see. What did Robert Hooke do? What did Robert Hooke do? He discovered cell. So P. Okay, Robert Brown, what did he do? This Robert Brown discovered nucleus. So cute. Schleiden and Schwann. Schleiden, not only Schleiden, but Schwann also, both of them gave cell theory. This is R and Camillo Golgi, as the name suggests, discovered Golgi apparatus. So P3Q4 R1 S2. So answer is first option. Okay, this was very easy question. Moving on to question 143. What does it say? It says the light reaction of photosynthesis does not produce which of the following? Yes, the light reaction of photosynthesis does not produce sugar because sugar is produced in dark reaction, which is nothing but Kelvin cycle. Okay. But Kelvin cycle. Okay, I hope it's clear to you. So, in light reaction, ATP, NADPH, oxygen, high energy of electrons, everything is produced, but sugar is produced in dark reaction, which is Kelvin cycle. Okay, now I hope that is clear to you. Moving on to question 144. What does it say? It says euglenoids have protein-rich layer of cell wall, which is known as pellicle. Okay, so this is a direct NCERT statement and you should know this. So again, match the following. We have to match this 145 question. What does it say? Chlamydomonas. Okay, so there are three options. Okay, Volvox is a colonial algae. Volvox is a colonial algae. Just a second. Volvox is a colonial algae. Spirogyra and Urothrix. Spirogyra and Urothrix are filamentous. Spirogyra and Urothrix are filamentous. And Chlamydomonas, as we all know, is a unicellular algae. So we have A2B3C1. A2B3C1. That is second option is the correct answer. Moving on to question 146. What does it say? It says multicellular and branched rhizoids are found in sphagnum. Okay. Sphagnum. Volvox and Cedrus, they do not have. Okay, just a second. So, Volvox and Cedrus do not have branched rhizoids. They have branched stems. Okay. So, answer to this question is option number 1. Moving on to question 147. What does it say? Algal general, which are Okay, so they want to ask which of the question, this is a spelling mistake. They want to ask that which algae are haplodiploidic. Okay, so answer to question 147 is Ectocarpus, Polysiphonia, and Kelps. Ectocarpus, the algal genera. This is a mistake. This is algal genera, not algal general. Algal genera, which are haplodiploidic, are Ectocarpus, Polysiphonia, and Kelps, and this is in direct NCERT statement. So answer is option 4. Moving on to question 148. It means haplodiploidic means they have both haplontic and diplontic stages in their life cycle. Okay. And both of the stages are equally occupying their life cycles. Okay, neither haplontic nor diplontic is dominant. Both of the stages are equally occupying their life cycle. That is why they are haplodiploidic. Okay. So moving to question 148. The leafy stages in mosses develop from secondary protonema. Okay, so mosses have a structure called protonema. From these protonema, leaf-like structures in mosses develop because mosses are bryophytes. So they do not have leaves. They have leaf-like structures and they develop from protonema. Okay. In which of the following lateral branches originate from basal and underground portion of main stem, grow horizontally beneath the soil, and then come out obliquely upward, giving rise to leafy shoots? Yes, so this is a modification, plant modification of stem. Okay, what are these? These are, see, just a second. Yes. So they are leaf. See, what they are saying is there are see, they are saying is, so this is a underground portion of main stem. From underground portion of main stem, lateral branches originate. They grow horizontal beneath the soil and then come out obliquely upwards, giving leafy shoots. Okay, so this is what happens. And this kind of thing is seen in chrysanthemum, banana. See, there are other examples also. This kind of thing is seen in banana, pineapple, and chrysanthemum. Okay, I hope that's clear to you. So these are the stem modifications we see. Okay, moving on to question 140. It is an assertion reason type question. Assertion is given and reason is given. Both apical and intercalary meristems are primary meristems. Yes, it's incorrect. It is a correct statement. Both apical and intercalary meristems are, they are primary meristems. Okay. That means these meristems are not, primary meristems, it means they have not undergone differentiation. Okay. They appear early in life on plant and contribute to formation of primary plant body. They appear early in life of plant and contribute to formation of plant body. Yes. So both apical and intercalary meristems are primary meristems because they appear early in life of plant and contribute to formation of primary plant body. So assertion is true, reason is true, and assertion is correctly explaining the reason. Oh, sorry, reason is correctly explaining the assertion. Okay, apical and intercalary meristem, they do not undergo differentiation. They appear early in the life of a plant and contribute to formation of primary plant body. So answer to this question is both A and R are true and R is correct explanation to A. So answer is option number 4. So I hope this was clear to you. This is the explanation for answers of your botany section. And if you have any doubt, put them in comments. So see you till next video.